Okay, thank you for having me this evening. 20 minutes is not very long, so I've got to rush through some of these slides. I might even skip a couple. We're an AIM-listed company, roughly 70, 80 million market cap, depending on the spread, 51 million of net cash. Based in London, but with R&D in Berlin, and our building never looks like that because it's never that sunny in Berlin, so that's an unfair representation already. And I guess the story of Silence Therapeutics is really one of precision medicine, and precision medicine is something that's talked about a lot. I think it's fair to say, obviously, things are getting more precise, but today's medicine is probably anything but precise in some respects. Kind of zooming into precision medicine and the concept of precision medicine really started with the Human Genome Project, which was a project started in 1990, took about 12, 13 years to finish, cost about $3 billion, and essentially it was an effort to identify, a successful effort, identify and map every gene in a human genome. So as I said, that cost $3 billion. I think today, if you went to the right places, you could have your individual whole genome sequence probably for something in the region of $1,000, $2,000, depending where you go. And that would, in effect, be using a machine of a company called Illumina. There are other players in this field. And you'll probably walk away with a memory stick, and the memory stick will literally have 3 billion letters in a text file. It's probably quite hard to store even in a memory stick. And it literally will be a continuous, just a text file of a jumbled up set of A's, G's, T's, and this is basically your entire genome separated into sort of individual genes. The next stage of that is, someone's got to actually analyze this data, and that in of itself is quite a gigantic task. It's only 50,000 whole genomes probably have been sequenced to date so far in the world, so there's not that much precedent out there. But ultimately, where we sort of enter the fray is really you have to have a therapeutic toolkit, if you like, to basically be able to, I guess, become a precision medicine player and take advantage of this type of explosion of data that's going to be sort of coming more and more. And there's a variety of different tools to be able to do that, and I'll go through our particular capabilities. One particular, if you like, approach is the new gene editing, which is a very hot area of CRISPR-Cas9 gene editing, and I'll come into that. But essentially, whatever you want to do, you want to switch on a gene, switch off a gene, edit a gene. One of the big barriers to entry, which I think the predecessor before said discussed, is you need a delivery technology to be able to do that. So to get into a cell intracellularly from out to in with any kind of, let's call it gene-modifying agent, you need specialist delivery systems. And really, essentially, at the heart of a technology company such as ours, delivery technologies is really a very, very important part of our offering. This is, again, this was called the central dogma of biology. DNA is transcribed to RNA, then translated to protein, and it's really a graphical representation of where we sit at the RNA level. At the DNA level, you've got the kind of traditional gene therapy companies, and I guess one difference, there are very many differences, is really a question of permanency and transiency. So when you go into the DNA and you start editing it or using gene therapy, that's a permanent effect. Permanent effects are nice as long as you're absolutely sure they're safe and they work. If they don't, permanency is not such a good idea. Transiency has the benefit of a better safety profile, so you can maybe do some repeat dosing and not be so dramatic in your effect, but obviously then you get into this world of chronic dosing and having to keep redosing the patient. So, you're in that sort of world between permanency and transiency. Very quickly, a very quick piece of cellular biology in the most simple terms that I could think of. In the middle here, we've got, if you like, a healthy cell. Within every cell in every human body, there's a nucleus, and within the nucleus, you can think of the entire DNA in every single nucleus. Think of DNA as an encyclopedia. Each chapter is one gene, let's say. So a gene is a particular chapter. Let's call it chapter 1, gene 1, 2, 3, 4. And the cellular process of life, if you like, is a cell needs to make a protein. It goes and finds the relevant chapter. So let's say chapter 1, it's got a certain code to make a protein. It extracts that chapter 1, and it copies it onto, let's call it a temporary structure called a messenger RNA or mRNA. So it's taking it away from the original source, putting it into a temporary structure. And this code is depicted by the different colors here. At this stage, the cell, one of the cell machinery systems called the ribosome will start reading this code and start making proteins. And this is basically the circular process of life, the encyclopedia, the genes. Each gene has a specific code for a certain protein, and the cell just goes round and round doing this kind of functionality. And I guess you can think of diseases in two different ways. There's either too much of a gene expression, if you like, or too little of gene expression. So think of it as a, I don't know, let's think of it as a sort of music stereo system, and you know the bars that are going up and down with volume, that's what we call kind of gene expression. So in the example on the left, where we've called a disease an upregulated gene expression, let's consider something like cancer. This whole process of DNA, messenger RNA, protein is replicated again, but as you can see, there's too many messenger RNAs being made there. So somehow, let's say a stop function is deleted or not quite working, and this is like a sort of stuck record, just constantly making proteins. You know, maybe an example of this, as I said, is in oncology. It's not a great example, but let's just use it for the moment. So, you know, this constant overactivity is something which is interesting to us. So here you'd like to switch this gene off, if you like, and let's say tone it down, maybe even switch it off entirely. And this is a key sort of point in genetics, is the basis of what we do. And if you like, ultimately, I think precision medicine is a very natural concept of what's called Watson-Crick base pairing. Genes are codes, and in a cell, opposites attract. And if you can make the exact opposite code to a gene, you can start having a therapeutic effect. So going back to the Human Genome Project, we know the code to every gene, as does everybody else. So what we can do is externally synthesize an anticode to every gene in the human genome. And that's called a short interfering RNA. So in effect, we make the anticode to the gene that we want to target. We put it in one of our specialist delivery systems. We get intracellular entry, we get into the cell, and on the basis of opposites attract, like I said, anticodes and codes coming together, the short interfering RNA, which is the exact opposite code to a gene, will match. This matching process is basically noticed by the cell machinery, which basically knows that it has to start cutting the gene up. This cutting procedure is really what gene silencing is. It's, if you like, what's called RNA interference, the RNA interference pathway, and it really is this kind of Watson-Crick base pairing and opposites attract. So that's what we do when we turn genes off. On the right-hand side is a very extreme example of the encyclopedia, where sometimes a chapter is missing. Take a disease like cystic fibrosis. A gene is missing, so by definition it can't be copied to the messenger RNA, it can't make the protein, so there's just a total underactivity, which is obviously can be devastating. In this instance, again, we use the Human Genome Project as our template. We can synthesize and create every gene externally, so we put it in our delivery system, get intracellular uptake. The mRNA will, if you like, hey presto, light up in the middle of the cell, and that's a cue for the cell to start reading it. The ribosomes will start reading it and creating protein. So at this stage, you know, we say that Silence has got the capability in certain cell types and tissues to be able to modulate gene expression on and off. And the specificity, if you like, and the precision medicine angle is because genes have a specific code, and the anticodes to that are highly specific to that. So that's kind of, if you like, the very beginnings of what you can think is really very precise medicine. Okay, so I talked about delivery technology, not to spend too much time on it. On the right-hand side, we have a variety of different systems which target a variety of different organs. Actually, nowadays, we've gone past the stage of talking about organs as such. We've zoomed in even further. So we're interested in specific cell types within specific organs. So we have a lot of delivery systems. We've got specific ones to the lung endothelial systems. We have specific ones to the liver and hepatocyte cell site systems. And in effect, these delivery systems carry gene modulating cargo or payload, whatever you want to do with it. There are a variety of different reasons why you need those. payloads are basically, they have negative charges, all sorts of different complications. They're immunogenic when they're kind of not protected by a delivery system. But really at the heart of any kind of, as I said, precision medicine is a delivery system. Okay, sorry, I'm going to skip this. Okay, so it's worth spending a bit of time on this slightly convoluted slide, but our drug, if you like, is a two-component system. As I said previously, it's a delivery system, and let's say for this example, a short interfering RNA. So on the left-hand side, we have a delivery system that's very awkwardly and badly named DAC, but that is our lung-specific delivery system. So one proof of concept is, if you like, we do a biodistribution. The biodistribution, as you can see on the bar charts, we're predominantly and mainly seeing the short interfering RNA end in the lung. So that's good. That means there's a variety of different lung indications we're interested in. Importantly, we're not ending up in too many places, although there is a bit of distribution in the liver. Sometimes you only want to specifically silence something in the lung and not have too much impact in the liver or the pancreas, etc. So that's a nice proof of concept which shows that we're ending up with our delivery system where we want to. And on the bottom side, we have what we call, let's say, the efficacy, right? So as a good example, we've got here, we take a disease, let's call it pulmonary arterial hypertension. The target gene here is called ET1, call it one of the chapters in the DNA. We've chosen ET1 because we've done a lot of work into it. There's a lot of literature that says that ET1 and overexpression, that example I gave where there's too much of ET1 being made, is potentially causal and active in PAH as a disease. So we use our lung delivery system we create the anticode or short interfering RNA to ET1 because we know the code to ET1. And in effect there, the bar chart on the left is ET1 is a silencing effect, the 81%. So here we're showing that the short interfering RNA is reducing the expression of ET1 by 81%. So there's a silencing effect going on. This is a mouse model. So that proves as a nice early efficacy marker. That's basically, in effect, if your hypothesis is right, that ET1 is problematic in PAH, you should be able to have a disease benefit there. So different delivery systems of different diseases require different approaches. In the middle, we've got example of a liver problem called liver fibrosis. Our target gene here is one called FAS. Obviously, we know the code to FAS. Again, take biodistribution, you see we're ending up in the liver a lot. And you can see the same sort of silencing effect. I can't actually see what the percentage is, 88%. And again, moving to the next one. Now, Antiplex is a much broader delivery system, much more systemic, so we do want to end up in a lot of different places. This is probably particularly useful for metastatic cancer, where you want to silence in not just a specific location, but everywhere in the body. Here we're given an example of a target gene called PKN3. Again, you can see that same process of some efficacy with the silencing. Some people ask, what's the right percentage, if you like? Is it 99%? Is it 60%? And that's a difficult question. It varies from target to target, disease by disease. But sometimes the more the better, I guess, is a good starting point. The flip side of it is, again, when there's a gene missing, like I said, there's a chapter missing in the DNA, we have the same sort of concept. So let's take the lung delivery system again. We're showing the proof of concept that we're ending up in the lung. On the efficacy side, and I think I should point this out, this is a mouse without a gene to make EPO. So you can see, obviously, the protein production is nothing. When we go in we replace the gene with a delivery system, mRNA, you can see suddenly after 6 hours, 12 hours, and 24 hours, we're seeing a tailing off, but a huge production. And that's a proof of concept that the mRNA is ending up in the right place, being read by the ribosomes and protein being created. So that kind of translates to what we call our genetic toolkit. We take a variety of different payloads, whether it's siRNA to switch a gene off, messenger RNA to switch a gene on. We can even and have got data in the CRISPR-Cas9 system to edit genes. We mix and match it with our delivery systems depending on which cell types and organs we want to go to, depending on the disease. We add what's called the translational science side, and I come to what that is. And that really is what we call our development program or our platform to make drugs. So we are a company that is not trying to make a drug, we're trying to make a platform for drugs. And I'll come to why that's potentially interesting. Okay, so I'm getting close. Okay, so this is slightly unfair example of a difference between a small molecule and potentially an RNA, an oligonucleotide-based approach to drug development. And really at the back end, when there's a clinical trial, unfortunately we're all in this kind of very difficult go slow system. Drugs have to be tested in humans, you have to have a safety first and foremost, then you go into longer efficacy trials. So it's very hard, even although you can get some breakthrough designations, you can, if you're showing dramatic effect, the regulator will speed up your drug. It's quite hard to make a meaningful impact, though, in sort of human trials in terms of timeline. But where we do have a significant edge is in the first process when we're actually making the drug. So as I said, if someone here in the audience came out with a gene which they think is of interest for a therapeutic candidate, let's call it PCSK9, I could ring up the office tomorrow and within 3 months have the anticode against PCSK9 in a delivery system, good to go as a drug. And as an example, something in the same concept as a small molecule can take 4 to 5 years. So the drug element of our world, if you like, is very fast to make, and it's the same drug all the way through if you get it through to approval. Okay, so I've got 5 minutes, and I think I want to go to this slide first. So, I mentioned translational science. Another way to look at our company is technology, which is our genetic toolkit, feeds into what we call translational science, which obviously, hopefully, will make clinical products. And translational science in our world is really applying an algorithm internally and externally to basically find diseases which our genetic toolkit can address. So, this is a very simplistic way of how we look at things, but first and foremost, if you worked in our translational department, you'd be looking for a disease or an indication where there's dysregulated gene expression, so something that's whacked up or just missing or very low expression. And when you find that, you think, well, can I deliver my siRNA or messenger RNA or CRISPR-Cas9 to the affected tissue? So if it's a lung disease, like say cystic fibrosis, does my delivery system get there? If it's a brain disease such as dementia or Alzheimer's, we're going to struggle because we can't get to the blood-brain through the blood-brain barrier. And this is a hypothesis that we just keep working. Do we have a competitive edge over other people? Are there any early signs of efficacy? So we're really sort of building up an argument to try and find something to basically go and target. And what that translates to is this really is what we call our R&D engine, and this is really the business plan of the company. And what I said earlier is we're not trying to make a drug, we're trying to have a platform of drugs. And you can see here a couple of things to point out. There's some timelines. We think from the moment we have an idea to potentially an IND filing or let's say commencement of a phase 1, it should be roughly 2 years. It can depend. It can be probably between 2 to 3 years, which is extremely fast development. And there's a very simple set of processes we take, and really they're kind of quantified in these 4 boxes. In Q1 of the idea, once we have a target and we have a disease which has been worked out by the translational science team, as I said, we go and synthesize a short interfering RNA within a delivery system. That takes 1 quarter, 3 months. And then we're straight into kind of animal models of efficacy to try and work out whether we can, we try and mimic the human disease, try and find animal models which mimics human disease, and try and see if silencing that particular gene that our hypothesis has sort of brought up is having disease impact. If that works, we then basically go past the red line where things start getting more expensive. You have to go into non-human primates and start looking at the toxicity profile of your drug. We start thinking about making clinical drug material and actually going into humans as in the yellow slide. So you notice that these dots are really some of our, at the moment, best ideas that we've worked on. And in a way, you can see we're pretty disease agnostic. We're not neither a cancer company or a metabolic disorder company or a lung company. And that's primarily because, as I said earlier, we're looking at diseases almost in a different way. We're looking at dysregulated gene expression, too much or too little of something. Can we deliver there? And then almost from there, we go to the disease rather than from the disease to the gene, if you like. So, the idea here is what we call our multiple shots at goal and high throughput business plan. The idea being that we want to populate this screen more and more. We want to be in a situation where we can have 20 to 30 ideas, which may or may not score within our sort of ranking system of what's a good drug and not. And rather than having, a traditional way of having one candidate, having to live with that, going ahead with that and all the risk that comes with biotechnology, we want to basically have this many-to-one relationship. So 20 preclinical ideas, the idea being that, at the end of 2 years or whatever, you can sit down in a room and say, look, out of this 20, 12 of them have failed, 3 of them are acceptable, 5 of them are potentially very good, maybe 2 of them we can go forward with. So that, you can have this kind of, for me, a backup, almost insurance policy system, where you can really have a more kind of smoother business than this kind of crash bang wallop that can happen with biotech stocks once drugs, have a problem in clinical trials. So, in conclusion, it's highly specific. It's the beginnings, and we're actively in the world of precision medicine. And I guess I'll leave one, when I was talking to one of our oncologists, sort of scenario that I think we want to get to in, let's say, oncology. You go to, take the example of prostate cancer, with the onset of things called liquid biopsies, where you can have a simple blood test for cancer, and that blood test will detect which particular gene is mutated. At that moment, because cancer is obviously mutating all the time, the oncologist can turn around, maybe to a company like Silence Therapeutics, and says, my patient has got a mutation in this particular gene. Could you go and synthesize your siRNA as quickly as you can to this particular mutation? We will be able to do that quickly, and that will be the beginning of really genuine point-of-care kind of precision medicine. Okay, thank you.
Questions?
Very interesting, thank you very much. One question. Just managed to slip my mind again. I'll come back to me in a second.
It's all right, don't worry, I haven't.
Put myself off.
I nearly forgot what I was going to say.
Anybody else got questions? Gentleman at the front.
Could you just explain a little bit about the delivery system? I got the bit about the anti-RNA, but I'm totally nonplussed about how you're actually getting it, one, into the cell, and then presumably you've got to get it into the nucleus.
Well, so actually we don't, first of all, go to the nucleus. We actually go to the cytosol with the sRNA or the messenger RNA. So that's one advantage, if you like. The nucleus is, for CRISPR-Cas9, we do need to get into the nucleus. The other thing I would say is you're unearthing a Pandora's box of nightmares when you want to talk about delivery systems. But we have a variety of them. Historically, we've been working with lipid nanoparticles. So that's what has been quite successful. There's a world where in siRNAs, lipid nanoparticles are becoming, let's say, less fashionable. For messenger RNA, when putting a gene back, they are an absolute must. We are using now what's called a conjugated system for siRNA delivery. And that is very specific, very successful, albeit restricted to hepatocytes in the liver. But we're doing very well with that, and that's a kind of industry trend towards that. Lipid nanoparticles are difficult. You've got to, you get limited by more acute conditions. Anything which is chronic and long-lasting, lipid nanoparticles can have a toxicity profile. They're not benign delivery systems. The conjugation approach seemingly is very, very useful for chronic conditions where you can repeat dose.
And so that's chemically conjugated, is it?
Yeah, so conjugation is called a GalNAc system. The sRNA is chemically modified, which is attached to it via a linker. And it targets a particular receptor on hepatocytes, Asia-glycoprotein, which is highly expressed, so we know how we get in. This works very, very well.
Could I assume for lung delivery, one of the tricks you'd use is actually physically deliver it to the lung via a nebulizer or inhaler?
I mean, we've tried. Some people say they've had some success. Any of that kind of stuff, the uptake is very little in our hands anyway. So we've not really seen too much success with that. Although some competitors with CRISPR-Cas9 with cystic fibrosis have shown some data. I'm not sure I'm entirely convinced. Right?
Questions, anybody else?
In terms of the degree of precision and personalisation,
right.
It seemed what you're describing was that the mRNA sequence that you're locking to seems to be specific to the disease rather than to the patient, or is there a combination? So when you have a patient in a trial, are you customizing something for their specific mutation, or is it more generalized lock to the disease time?
Well, look, first of all, some of this stuff is not of today necessarily. So so first of all, you're talking about cancer. That's something different to, let's say, a metabolic disorder. Right? So metabolic disorder, let's say hypercholesteremia, we're generically targeting PCSK9, right? So it's not necessarily patient to patient. In oncology, in time, although not necessarily now, and it will be on an individual patient's tumor, which is obviously mutating fast.
You don't have to deal with the head of having to do personalized work to customize it for an individual candidate.
Yeah, I mean, the idea being, look, a short interfering RNA is, theoretically anyway, a laser-guided missile. It's highly specific to the gene you target, right? If you get the wrong gene, it's just going to fire empty. It's just, a dart going into empty space. But if it locks to the target messenger RNA, i.e. you got the right mutation, if you get into the cell type, if you get into the tumor cell, then it's potentially, and we get into another world of chaotic genomes and things like that. But theoretically speaking, we're always trying to lock into that specificity.
Gentleman towards the back.
What is the company's financial position at the moment?
So we have last reported about 51 million pounds of cash. We spend about 10 million. I mean, obviously, if I can just, I can't.
We'll just set it. Yeah, you can now. Yeah.
Sorry. One of the good things and bad things is that this particular slide is preclinical data. So, if I was to do a chart of costs, to be honest, this is pretty cheap and cheerful, and everyone is in a very good mood here because you can get a lot done very fast and very cheaply. Once you start getting here, it's like this, and then it just gets exponential. So there is a point at which, things will start changing materially in terms of cost. Hopefully by then, we have this many-to-one relationship where we can take our best high-conviction ideas. The idea is to have very high-conviction ideas. So, to try as much as possible to reduce that human clinical trial risk, which happens a lot, I mean, if you believe in this kind of concept of Watson-Crick base pairing, it's a little bit far-fetched, but theoretically speaking, from the very beginning with a mouse, if you really got it right and your biology is right and you really have found the right gene and you really have got the right delivery, theoretically that should translate all the way through to humans because you're doing something which is very specific rather than something kind of generic, if you like, and systemic.
What sort of revenues would you expect from major drug companies?
Don't know. I don't know how to answer that. Anything from nothing to multi-billions, if that helps.
I'm on your ATO-027. That's now in clinical, is it still
so
preclinical?
ATO-27 is in the clinic. It's just had a phase 2A readout. So we're in humans too. It's a drug which we're thinking about. Again, it comes down to this cost. Do we want to spend 20 million pounds to go into a full randomized control trial with pancreas cancer, or do we think we can do better with this business plan? It's in the balance.
Jury's out.
Well, ATO-27 is an 11-year-old drug. It started in 2005, right? So I don't know, what was the last technology you used from 2005? There is an element of that, especially with our new delivery systems. Incredibly potent.